of my studies in psychology and neuroscience, I've always been interested in the link between anxiety and depression. So for this study, I started looking into a diagnosis called anxious depression. And I found in the literature that 74% of people with a major depressive disorder diagnosis also have anxiety disorder diagnoses. And this comorbidity leads to a greater risk of functional occupational impairment, decreased response to treatment, and a more chronic course of illness. And conveniently, Professor Fake's lab is in a neuroimmunology lab, and I found also in recent literature a possible link of the immune system, and we'll get into that later. So first, I wanted to talk a little bit about the behavioral immune system. So we're all used to the common symptoms of the common cold, coughing, sneezing, etc. But what about the behaviors that you exhibit when you're sick? So when we experience an immune threat, either a vaccine or the common cold, we might experience sickness behaviors like decreased appetite, irritability, decreased sociability, increased pain sensitivity, and depressed affect and anhedonia. So when we have something like the common cold or an acute illness, these behaviors can be incredibly adaptive. Temporary social withdrawal might mitigate more immune risk from other people. Behavior changes can cue or close others to help you out, maybe bring you some soup. And lethargy allows the body to allocate energy to fighting illness instead of other things. But when we consider chronic illness, these behaviors might turn into things like depression, consistent low appetite, persistent sadness, poor sleep and chronic low energy, and loss of interest in pleasurable activities. But I wanted to expand everyone's perceptions of maybe from physical chronic illness to also mental chronic illnesses. I found in the literature that anxiety, specifically attachment anxiety, I thought this could be a good way to study persistent anxiety in a population like Hamilton. And attachment anxiety has been found to predict systemic inflammation. And also, depression symptoms are correlated with attachment anxiety and systemic inflammation. So in my study here, I'm proposing a model of attachment anxiety causing inflammation, which might cause depression symptoms. So for those of you who are unfamiliar with attachment style, attachment theory suggests that people differ to the extent in which they believe close others will be supportive and available during times of need, depending on their early life experiences with care providers. And there are two types of insecure attachment. Anxious attachment outlines intense fear of rejection. Those people are usually dependent on others and they worry about their close relationships. And people with avoidant attachment are excessively self-reliant and pretty uncomfortable with closeness and intimacy. Another big thing implicated in attachment insecurity is social withdrawal. Heightened inflammation has been found to encourage social withdrawal, as we discussed before with sickness behaviors. Insecure attachment tends to discourage healthy support seeking, and anxiously attached individuals might consider themselves unworthy of help. So I wanted to look at people's healthy support seeking nature during times of sickness. But this is a neuroscience thesis, so let's talk about the brain. When we experience a stressor, either general or social, which might be more implicated in insecure attachment, our stress circuit is activated. This is called the hypothalamic pituitary adrenal axis, and two of the main stops in this circuit are called the hypothalamus and the hippocampus. The hypothalamus receives signals from higher brain structures like the amygdala, which trigger fear responses, and the hypothalamus is responsible for kind of triggering the body's response to stress. And the hippocampus is a negative regulator of this circuit. So perhaps we've seen something scary before and we know not to be scared. The hippocampus would tell our stress response to calm down a little bit. And conveniently, these two stops in this circuit are the main sites in the brain for pro-inflammatory cytokine synthesis. So what the heck is that? Pro-inflammatory cytokines are proteins synthesized by activating accessory immune cells in response to an immune threat, like a vaccine or sickness. Increased synthesis of these proteins leads to inflammation, and they can be produced in the brain as well as peripherally in your body. Peripheral cytokines may activate afferent nerves, cross the blood-brain barrier, etc., such that the brain and body are in communication during times of sickness. And when experimentally administered, subjects will usually display sickness behaviors. So introducing these proteins experimentally will cause kind of the natural symptoms of sickness. So getting into my hypotheses, I thought that those endorsing high attachment anxiety would display a larger increase in inflammation in response to immune threat, endorse more social withdrawal behaviors after a vaccine, and be more likely to endorse depressive symptoms during an immune challenge. Getting into methods, I first wanted to talk about why we chose to do a vaccine study. Vaccines are pretty commonly used in lab to induce sickness because obviously we can't actually make people sick. So they're a pretty consistent and ethical way to do so. And there are no existing studies looking at attachment style and immune reactivity, so I thought it could be interesting to look into that as well. So my demographics, I had 36 Hamilton College students aged 18 to 21, 26 were female and 10 were male. I had a majority white American sample. For our sessions, we had participants come in twice. once at baseline, so before they had had any vaccine, and a second time 4 to 6 hours after they had received a flu or COVID shot, or coming in for a placebo, which was a fake saline nasal spray, which we told them was an immune booster. No one was discouraged from actually getting their vaccinations. Then participants provided a salivary sample as well as their heart rate variability through an electrocardiogram, and then they completed some surveys. So our main marker for inflammation was meant to be obtained through salivary IL-6 measures. IL-6 is one of those pro-inflammatory cytokines I was talking about earlier. But unfortunately, we experienced some setbacks in the biosafety department, so we weren't able to assay this data. So instead, we focused on parasympathetic nervous system activity. This was collected through the Mindware Biomix data collections system, as shown here. And we collected, again, a baseline electrocardiogram. And this measured people's high-frequency heart rate variability for 5 minutes and decreased heart rate variability or parasympathetic nervous system activity was associated with elevated IL-6 levels or higher inflammation. And then the surveys that people completed were a depression scale to determine their depression changes from pre-vaccine to post-vaccine, the adult attachment questionnaire to determine their attachment style, and the sociability scale to get into those social behaviors that I was talking about before. So for results, for my statistical modeling, my independent variables were grouped, so whether people received a vaccine or a placebo, and attachment style. I just wanted to note that for ease of graphing, we split people into low and high categories for each type of attachment style, and that was done by taking all of our data, getting the median for anxious attachment and avoidant attachment, and anyone above the median was considered high, and anyone below the median was considered low. And my dependent variables were PNS activity, general sociability, and depression. So first, I just want to orient you here. Below this 0 line here, people were showing decreased inflammation, and above that line, decreased inflammation, and below the 0 line, people were showing increased. So this is an inverse relationship, as I said before, and we did not find that the vaccine reliably impacted PNS activity. So first, with that in mind, here again we have average PNS activity on the y-axis and attachment anxiety on the x-axis. And we didn't find any significant results in the attachment anxiety group, but however, we did find a significant result of group and attachment in the low attachment avoidance group. So at first we were unsure why this might be, but we thought the placebo effect might have actually been a stress response because our placebo participants were concentrated towards the end of the semester around finals time. But again, I see you're perplexed, very interesting result. I'm open to interpretation. Next, we looked at general sociability difference from pre to post vaccine. So people were showing increased sociability and decreased sociability here on the y-axis, and then we've got attachment on the x-axis again. We didn't find anything significant in the attachment group, but again, we found this huge placebo difference in the low attachment avoidant group. We again talked about this a lot, and we thought that it may be a protective effect of vaccines such that people with this low cognitive avoidance might feel more inspired to go out and socialize after they receive the vaccine. And maybe people with low avoidance are more likely to engage in healthy support seeking. And lastly, we looked at depression difference. That's here on the y-axis, and we've got attachment on the x-axis again, and we didn't find any significant results in either the attachment anxiety or the avoidance group. So our depressive symptoms were not reliably induced by vaccination here. In summary, vaccines did not reliably induce inflammation as expected. Inflammatory stressors may encourage social help-seeking behaviors and insecurely attached individuals. And attachment avoidance is pretty understudied in this context. I didn't really find any literature talking about attachment avoidance, and I think it could be something really important to add to this literature. So for discussion, I did want to note that even though heart rate variability was found to be a strong proxy for inflammation, we did have some collection difficulties with this data as well. We lost a couple of participants to fuzzy data and things like that, so a couple of people didn't meet our exclusion criteria. Also, vaccines and actual illness aren't the same. Vaccines cause a lot less inflammation than an actual cold or sickness would. Also, this data grouping here, I wanted to go back a little bit and note that because of our smaller sample size, a lot of people in the high attachment anxiety group are the same people that are in the low attachment avoidance group. But we did have some people in both categories. I really wanted to create a secure attachment group, but unfortunately not a lot of people endorsed that. So that could be something to look into in our population. And finally, self-report is subject to bias, as you know. So for some future directions, of course, I would love to assay our salivary data and see maybe what a more direct assessment of inflammation would give us here. I'd love to investigate chronic physical illness to see maybe how attachment style interplays with existing high inflammation. Complete clinical assessments, so maybe have people with clinical diagnoses of anxiety, depression, and insecure attachment, and have a larger, more diverse population with those relevant diagnoses. Thank you everyone so much for coming. Thank you to my family, friends, and mentors. I would not be here without you. Questions? Yes, Dennis.
Are you at liberty to divulge? Oh, sorry.
You can go first.
No,
Or Professor Ford.
I was like, oh, please tell us what happened with the biosafety thing.
There were some setbacks with us getting vaccines and all of our stuff into that department, so we unfortunately weren't able to assay that data. But that's okay. We did collect it, and I hope it gets used at some point.
Maybe assay it soon.
Oh, okay, okay. It wasn't like a. Okay, I was like, there was a biosafety problem.
No, we didn't do anything.
There's a very protective biosafety committee here.
Oh, got it.
There are some spit popsicles ready for analysis somewhere.
Okay, alright, got it. Thanks.
Yes, Catherine.
You mentioned the ethics of like giving people illness, which like fair. And you mentioned that vaccines are very different from like a real immune response to illness. So I was wondering, is there a way that you could ethically like sample people who are already sick, or like do some sort of retrograde analysis?
Yeah, that's something I would be really interested into looking into. Maybe again, people with like physical chronic illnesses or things like that. Also, people sometimes for different disorders are treated with recombinant cytokines. So those are some studies that I've found in the literature as well. That would be, obviously we can't do that here, but very cool to read. Yes, Dennis.
Well, first of all, great presentation.
Thank you.
And I wanted to ask, did you, when you were surveying people prior to the testing, how many of the, or like, I guess like, did you take into account like pre-existing mental health issues and depression, or was that kind of
Yeah, we weren't able to ask people if they did have any clinical diagnoses, but we did also look at generalized anxiety and social anxiety and use those as controls. And those were all found to be highly correlated with attachment anxiety as well. Summer?
Do you think you might have had different results had your placebo method been different? Like if it was more believable, quote unquote?
That's a great question. Yeah, a lot of people didn't believe our placebo. Obviously, it's pretty clear that we're not able to give people an actual vaccine, and no one's ever really heard of the nasal immune booster before. But I'm not really sure who else would have done it. I thought they were pretty convincing, but I guess not. So that's a great question.